Hello everyone. So today we are going to talk about protein microarray. So let us begin. Uh, so uh, this protein microarray is widely used tool and technique in the field of proteomics, in which we study the proteome, which is an entire collection or entire set of all the expressive proteins at a particular condition, at a particular time, which is present in our cell, tissue, organs, and organism as a whole. In protein profiling, we look for how abundant a protein is, how it is interacting with other molecules, and what kind of functionality they are playing, what kind of activity they are doing, and what are the modification, and structural modification takes place in the protein. These all things come under protein profiling under proteomics. To do this task fast and accurately, we have certain techniques like MALDI-TOF, 2D gel electrophoresis, mass spectroscopy, and microarrays is one of them. So, the concept of microarray was came from theory of ambient analyte immunoassay, which was given by Roger Ekins in 1989, which was later transformed into DNA microarray. And we all know that this DNA microarray was the base of genomics. It was a boon for the genomics field. But due to some limitations in the DNA microarray, the protein microarray was developed to overcome these limitations. Limitations were like, firstly, you cannot correlate the messenger RNA which is present in the cell with the abundance of the protein it will produce. You cannot correlate that. Now, secondly, the post translational modifications. Now these are like, for example, methylation, which is addition of methyl group, acetylation, which is addition of acetyl group, and like ubiquitination, which is degradation of proteins. So these post translational modifications, they ultimately changes the stability, the folding, the structure, the compartmentalization or localization of protein, like when after they are manufactured, where they will get localized and which organally. So these PTMs are of huge importance, which cannot be studied using DNA microarray. So protein microarray of huge importance, like they are high throughput method, which means that they can analyze large number of analyte, or large number of molecules in your very small sample in a single experiment. If you are taking a very small quantity of your sample, then they have the ability to analyze the entire molecules which are present. So they are high throughput method. Then these are rapid, they are fast, they are automated, and they are very sensitive as well. And they consume a very small quantity of your sample, and they allow you to determine the function of these proteins on a larger scale. So the basic principle behind the protein microarray chips is that you take a support system which can be a slide, then you allow it to immobilize, you allow the proteins to immobilize on that slide, and then you design a probe. And once you design a probe, the important thing in that is you have to do the labeling, which can be fluorescent labeling, radioisotope labeling, or there are various other methods. So once the labeling is done, now you will allow it to incubate. So the antigen and antibody reaction will take place between the complementary molecules, and then there will be fluorescence generated. So before the fluorescence generated, you will give a wash, so the unbounded molecules will be washed off, and only the one which have been binded will fluoresce, and they will emit some kind of signal in the form of color, which can be detected. And they are, once they are detected, the data is generated, and this can be interpreted via by the expertise in the field. Now, how to make a microarray? So, in manufacturing, firstly, microarray chip fabrication is made, in which you have to take a support, which can be a glass slide, that can be a silicon slide, or micro titer plate. On these slides, you have these grooves, major grooves and wells, we can say, onto which the immobilization of protein is made. So the next step is protein spotting, in which you can take the help of robotic spotting. Then inkjet spotting methods are there, piezoelectric methods can be used, then photolithographic methods are used for spotting of the protein. Now, once this is spotted, there will be immobilization of the protein on the slide. Now this binding can be covalent, depending on the, in the fabrication part, I forget to tell that you have to make that slide compatible by doing aldehyde coating or epoxy derivatized coating, so that the binding can be covalent, random, affinity binding. These types of binding you will do based on the application part, which kind of application you are looking for, you will go for that. Like if covalent bonding will be there, then irreversible um, antibody will be immobilized. So once the fabrication is done, I will go for spotting, and there will be binding. Protein will be immobilized on the slide, and then you have to design your probe, which will be fluorescently labeled using various type of dyes like Psi 3 and Psi 5. Now Psi 3 will give green color on fluorescence, and the Psi 5 will give red. Radioisotopes can be used. Carbon radioisotope like carbon 14, phosphate 33, phosphate 34, and you can use various enzymes like horseradish peroxide, liposomes can be used. And if you are not going for dye labeling, then you can do detect these signals via using various techniques like AFM, atomic force microscopy, or surface plasmon resonance spectroscopy. So once these probing is done, and then the after the binding, the fluorescence will appear. Now there must be some detection system. So various types of microarray scanners are there, or confocal laser scanners are there, which will detect these signals, and then they will generate some data, which will be interpreted by the expertise. So that is how microarray manufacturing is done. Now I'll discuss the three major types of protein microarray. The first one is analytical microarray, second one is functional, and third one is reverse phase microarray. Let us talk them in detail. So firstly, in analytical microarray, what we do is we prepare a library of antibodies, aptamers or aphid bodies onto the glass slide. So here you use the protein molecule as a bait for a specific target which is present in the cell lysate of the patient's serum. So you prepare a library of these proteins, and the cell lysate is allowed to spread. And if that particular antibody you are looking for, or you are going for any diagnosis purpose, and you want to detect any antigen or antibody which is present in the cell lysate of the serum, then you will allow it to spread, and binding will take place, and you will come to know that yeah, this biomarker is present. So this type of technique is used in the in uh, uh, biomarker identification for various disease, also for clinical diagnosis purposes. You can also do a comparison between healthy tissue like a cancerous one with the normal one, and you will come to know various types of tumor stages and everything through these techniques. And you can also deduce the environmental factors, how they are affecting the protein, like environmental stress factors. Excuse me. Now the antibody microarray is the most common one which is used. In the figure you can see that you can use antigen direct antigen you can also label, and you can use secondary, like first one is the in the sandwich one, which is widely used in ELISA method. There is a capture protein or antibody, and then the serums antigen have been allowed to bind, and then secondary, or you can say reporter antibody is there, which is fluorescently labeled, and upon specific binding it will fluoresce and emit some signal. So this type of technique, this type of sandwich and analyte labeling method comes under analytical microarray technique of protein microarray. So, now the second one is the functional microarray, which is very important, because in this you come to know about the various types of interactions which protein do with other molecules, like how a protein is interacting with other proteins, how it is interacting with the DNA molecule, and how it is interacting with RNA and phospholipid and other such small molecules which are present inside the cell. And how it is different from analytical one is that on the glass slide you have to array a full length functional protein or a protein domain. That means, like, if you want to study a particular tissue, a particular tissue's functionality, then you have to array the complete protein domain, pure protein domain of that particular tissue onto a glass slide, and then the interaction studies can be made. So it is widely used in the various biochemical reactions which takes place inside our body, to study them in a single experiment. Now the third one is the reverse phase microarray. This is very opposite of the forward phase, which was analytical phase microarray. Uh, in that case, we use bait molecule onto the glass slide. Here we use cell lysate from the patient onto the glass slide. We mobilize the cell lysate components on the glass slide, and then we design some target protein, which will which we want to find out that whether this kind of antigen or antibody is present in the cell lysate of the patient. So we will design that antibody, and we will allow it to react and bind with the cell lysates components. So in this case, like, if any protein have been altered due to post translational modification, they can be studied. And this is of huge importance, reverse phase microarray. Now comes the application part. So basically, majorly the application part is in diagnosis and proteomics. In diagnosis, you can come to know, you can detect the antigen and antibodies in the blood sample. You can profile the various types of biomarker, disease biomarkers. You can monitor the disease state, like in which state the disease is, and if you are going for any personalized therapy, then you can came to know that how effective it is right now or not. Then antibody characterization, secondly, in which you can go for epitope mapping, cross reactivity, specificity of a particular antibody, you can do. Then thirdly, in proteomics, in which protein expression profiling, that is how abundant a protein is inside a cell, how functionality wise, how it is working, and what kind of interactions it is doing. You can do that in proteomics. Protein functional analysis, we have already talked. Then treatment development. So antigen specific therapies for autoimmunity, cancer, allergies have been made, and small molecules can be used as a new drugs. So they can also be tested via using these protein microarray chips. And like, for example, any protein dysfunctioning is occurring in any biochemical pathway. We know that the enzymes are the ones which regulate any biochemical pathway, and these enzymes are nothing but proteins. So if any protein dysfunctioning takes place, we can modify it, we can alter it, we can remove it, and we like ultimately we can design any therapy for that disease or that particular pathway which is the cause of the disease. So treatment in field of treatment development, it is of huge importance. Now the challenges. So in the challenges part, there are certain challenges as well, like firstly, they do not have longer shelf lives, because to maintain the protein in their native in their secondary and tertiary structure, it is little difficult, because they require wet environment, aqueous environment. So uh, to maintain their proper structure, that is of huge importance. um, the second one is sometimes improper binding takes place, because when you are fluorescently labeling any these of the proteins, then these fluorescent dyes sometimes bind to the active regions of the protein, and it will hinder the binding the target protein with the active region of that protein. So the uh, because it is all about binding in protein microarray, and if there will be improper binding, then that is of no importance. Now the third one is they are a little costly, so they are yet to flood the market. And yeah, these are the challenges. That's it. That's all for the protein microarray. Thanks a lot, and if you liked my video, then please do subscribe to my channel, and please comment me in the comment section if you want such kind of videos. And thanks a lot.